All right, so the origins of this project were about 4 years ago, I guess. And at the time I was working both for BiodiversityWorks and for a nonprofit called the Betsy and Jesse Fink Family Foundation, which has done a lot of work with various island farms on sustainability issues. And in the course of some discussions with a farmer at IGI, I think it was Andrew Woodruff, who a lot of you know, we were talking about ways that biodiversity can benefit farms, obviously, in terms of providing pollinator services and pest control services and so on farms. And I think it was Andrew who said, well, what if we turned around and asked what can farms do for the ecosystem rather than what the ecosystem can do for farms? So that was really where we started with this. Can we find some ways to not just minimize the negative ecological impacts of farming, but actually use farms as a positive ecological force on the landscape? And that made really great sense to me because as a birder, I know that farms offer really, really good habitat for birds and offer extremely good birding. So we had some discussions and we came up with these goals for the project. We wanted to do just a general survey of what island farms were already doing to support invertebrates on the vineyard. And we wanted to develop some methods by which they could do more and do it feasibly, allowing for the fact that farming is kind of a marginal activity and that farmers are incredibly busy during the growing season, but do more to support invertebrates. And we focused on a particular interest in bees that are specialists in the pollen they gather. And I'll talk about a little bit of that as we go along. And the genus Bombus, which is the genus of bumblebees. And they just seemed like they were an important genus to study. And in a general kind of way, we wanted to build on previous studies of bees on Martha's Vineyard, including the really wonderful one that Paul Goldstein coordinated back in 2010 that some of you may actually have worked on as volunteers. We enlisted 8 farms to participate, and it was quite a broad selection of farms all around the island. And they're ranged in size, they ranged in how they run their operation, and they range even in how they are organized financially, with 4 of them being nonprofit entities and 4 of them being for-profit enterprises. And we enlisted their cooperation, for which we were extremely grateful. And I have to say that as valuable as this project was, the thing that I remember most about it is that it was incredibly fun for me. It was going to these wonderful places repeatedly, doing throughout the growing season and looking for bugs and poking around in the flowers and in the crops they were growing and in the cover crops and in our project plots and everything. And farms are just wonderful places. So it was a really terrific opportunity for everybody who was involved in the project. One thing that we did was we installed what we called project plots, and these were to test a mix of species just to see what effect they had on pollinators, and we hoped that they would produce some suggestions that we could use for enhancing pollinator activity on farms. These were small plots. They were only, a couple of hundred square feet, and we got help from the farms tilling them, and in some cases putting in some piping for watering them. And we looked at our resources and we really realized that we weren't going to be able to do a controlled and rigorously quantitative manipulative experiment here, but really we thought of it as a pilot project where we did some manipulations with these project plots, and then we just studied them intensively to see what we could learn using the techniques of really naturalists rather than scientists. So that's the perspective that we were coming from. We were busy little bees, you might say. 261 individual visits to the farms across the 3 years. We generally started taking data in early May and continued until late October when cold weather put the kibosh on things. We collected about almost 2,300 observations in iNaturalist, representing 283 species. We also did some collecting. I collected 269 specimens that represented 64 species. Most of those were bees. And that brought the species count up to at least 297. It was actually a lot more than that, I'm sure, because there are a lot of things that we simply couldn't identify precisely. And then finally, a third kind of data that we collected involved just visual observations and recording them on data sheets that we prepared specifically for this project. So we recorded the status of the pollinator resources, floral resources on the farms, the condition of the project plots, and then we recorded everything we could see identified as well as we could do it, just visually. So, you know, in the case of some things that we maybe only get 1 or 2 photographs of, those photographs would be in iNaturalist, but we might have noted in our data sheet that we actually saw 200 of them. So there were multiple sources of data that were all kind of cross-referenced and could be checked. It's a little bit cumbersome, and in particular, those site visit narratives were unwieldy to use, but there is a huge amount of data stored in them, and we drew on that in doing our project reporting and analysis of the data. I won't talk too much about iNaturalist, since I know a lot of you are users and some of you may not be terribly interested. But we set up 2 individual projects, one of which was for insects we found on flowers in the project plots, and one was for insects outside the flower plots. We only recorded things that were on flowers or had visited flowers, that we saw visiting flowers. And because of the way iNaturalist works, it's really easy for us to just spatially separate the data from any one of those farms. So if you want to look at the data from the project plots from Mermaid Farm, it's very, very easy to do. Or the insects from outside the farm plots at Beetlebung Farm, it's very, very easy to do in iNaturalist. So we found iNat to be an extremely powerful data aggregation and analysis tool. And of course, one advantage of iNaturalist is that it offers both human and artificial intelligence resources for identifying things. So it was a huge help for identification as well. We tried, I guess, 9 species of plants in our project plots. Some of these were native, some of them were non-native. They were all picked because we expected them to be attractive to either pollinators generally or to specific subsets of pollinators, for example, goldenrod specialists, or several members of the genus Melisodes, the bee genus that really like sunflowers. And we had kind of mixed results with them. The butterfly weed, the first year, all that we had shipped in from off-island, all died of some unknown pathogen. Very droughty conditions in 2002 pretty much subdued all of our clover, although it bounced back in 2023. So kind of mixed results, but in general, the plants performed about how we expected they were going to. And we were also paying a lot of attention to the flowers outside our project plots, and that allowed us to identify a very long list of plant species that function really well as pollinator plants. A good example would be chicory, which is a common roadside and agricultural weed on the vineyard, but it turns out to be a really good pollinator plant. Who knew? A lot of herbs are very, very good. Queen Anne's lace is a really good pollinator plant, especially for small wasps and flies. And surprisingly, members of the radish family, which grow as weeds widely on farms, turn out to be surprisingly attractive to pollinators. I will say that dandelions, although they've received a lot of attention lately in the popular press as being an early season pollinator plant, really didn't do much as far as this project could tell. They were visited only sparingly and only by the most common generalist species that already had lots of other things to visit. So if you don't like dandelions in your yard, feel free to cut them down. We did come up with some plant recommendations that I'll go over very quickly. These apply specifically to the farms on the island, but I think they're also very relevant for people who are doing native plant landscaping or pollinator-friendly landscaping in their yards, for example, through BiodiversityWorks Natural Neighbors program. The real home run hitter were goldenrod and native aster. And I'm going to say this multiple times, I'm sure, during the course of this presentation. Plant goldenrod. It is just a magnificent plant for pollinators. It attracted both specialized bees and also a lot of generalists. And it accounted just goldenrod, the two goldenrod genera, accounted fully for 20% of all of the insect sightings that we logged in the course in iNaturalist in the course of this project. So enormously powerful pollinator plant. Even though it didn't do well in this study, we're confident to recommend butterfly weed, Asclepias tuberosa, as a plant for pollinators. It blooms fairly early in the season and a lot of things visit it. And spotted bee balm, Monarda punctata, is an excellent plant also, particularly for wasps, and I'll talk about that a little bit further on. Cover crop species, crimson clover, alfalfa, things like that, the ones that flower are good pollinator plants if they're left to bloom on a farm. Obviously, grasses don't do much, like winter wheat, because they don't have flowers that pollinators are attracted to. But a lot of other crops are cover crops are pretty useful to pollinators. And flowering herbs, everything from dill and fennel to things like coriander and thyme, all of those herbs are visited heavily by pollinators, primarily generalists, but we like everybody here. So, and finally, sunflowers, particularly the more traditional varieties, attracted a lot of the most workhorsey kinds of pollinators, Bombus impatiens and western honeybees and so on. And also they did a very good job of attracting their target species in the genus Melisodes. The planting methods that we used, tilling and then mulching and then periodic light weeding, worked really well, but you might be able to do without mulching. I think farmers should feel free to experiment. Native plants generally did well once established, once they get through that difficult first year. But anything you can do to help in terms of supplemental watering or weeding, help them get established in that first season is welcome. I know farms may not be able to do that just in terms of the labor cost, but to the extent that they can, it definitely helps. Untidy is good for pollinators. The edges, the sort of unmanaged edges and waste areas of farms produced a lot of insect activity because of the floral diversity found there and the structural complexity that they represent. And as I mentioned, many of the weeds turned out to be surprisingly good pollinator plants. And finally, we recommend putting your pollinator plants into single species blocks to minimize interspecies competition. We learned this the hard way in this project. For example, Euthymia graminifolia, one of our goldenrod species, was much more aggressive than Solidago nemoralis, our other goldenrod species. And by the third year, where those two species had been interplanted, the Euthymia had pretty much eradicated the Solidago. So if you put things together, you run the risk of one aggressive species crowding the others out. A look at the various diversity based on iNaturalist observations at the various farms. And you can see that it varied somewhat. And we thought a little bit about what accounts for that. The total species, for example, ranging from a low of 68 to a high of 115. And I think a lot of that was, some of that was attributable to how the farms were managed. But I think most of it was attributable to the landscape context of the farms. In other words, what's around the farms. And I'll show two slides, and I'm going to come back to these slides later on because I think they're really important. But this is the Farm Institute down at Katama, and you can see that the area around it is primarily pasture and row crops and road and structure, and there really isn't much there that a bee would want or any other pollinator. So there just simply wasn't much in the area around our project plot. And you have to remember that bees or any other pollinator have a sort of finite distance that they're going to disperse from their next nest site in order to forage. So unless you've got diverse pollinators in the general area, you're not going to get diverse pollinators, particularly in a pollinator plot that you put in. And I will talk about that a little bit later on. And then in contrast, this is North Tabor Farm out in Shellmark, and we've got everything from a little bit of red maple swamp to some oak woodland to some nice untidy edges and a beautiful blueberry patch and some other resources there. And this is one of the higher diversity farms. So very different context. It really doesn't say anything about how well the farms are managed. It really tells you more about how the area around the farms are acting to support wildlife. So let's look at some of the bugs we found. And we'll start with one of my favorite groups, the flies, the order Diptera. And we found a really high diversity of flies on farms, members of 19 taxonomic families, which is a pretty extraordinary number, I figure. And that species count of 49 would have been much higher, except that flies are extremely difficult to identify from photographs or visually in the field. So a lot of species are undoubtedly lumped at the genus or even the family level. And three of the families were particularly well represented, and I'll talk about them a little bit individually. But I think it's fair to say that farms on the vineyard are extremely important habitat in supporting fly diversity, and that the converse is also true, that flies are ecologically very important on farms and to farms. The family Syrphidae, the hoverflies or flower flies, was pretty well represented and accounted for more than half of our fly observations in iNaturalist. Both native and non-native ones were found. Some of these, the ones on this slide, were all very common, and they're all non-native species. That makes sense that they would show up in agricultural environments where a lot of non-native plants have also been introduced over the decades and centuries. But we also found some pretty rare and unusual, apparently rare and unusual syrphid flies. I should say that just the fact that something has not been recorded previously on the vineyard doesn't really mean that it's rare, because nobody is looking at flies. Nobody has studied flies very intensively. But on the other hand, there are well over 50,000 observations from the vineyard in iNaturalist. And if a big fly like Malata posticata over there on the left hasn't been photographed by somebody previously, it probably is not a common fly. So I think it's safe to assume that these first records, these first iNaturalist records from the vineyard, probably were pretty significant in terms of documenting rare species. And again, that's a tribute to the farms. It suggests that they're very important and very high quality fly habitat. There's a lot of nutrient cycling going on in farms, things composting and breaking down and growing. And that cycling of nutrients is kind of the process that flies are really, really good at getting a piece of. A lot of them are what are called detritivores, meaning they eat decomposing material. And so, crops that have been plowed under represent food. Compost piles represent food for the larval stages of a lot of these flies, the adults of which then visit flowers. Californidae, the blowflies, are a family that are often detritivores. Most of them are detritivores, although one genus, Billardia, actually has larvae that parasitize earthworms. And Billardia also gives birth to their young live instead of laying eggs. I mean, who knew? This is stuff even I didn't know about flies, and it just shows how incredibly diverse the biology of flies is. Not a lot of diversity, but a lot of individual records of these genera. And then finally, Tachinidae, the bristle flies. This is a large and important fly family. And all of the members that I know of this family have larvae that are parasites on other arthropods. And we did not find a lot of diversity of tachinids, but we found large numbers of a few genera, and they turn out to be really useful genera. One of them has larvae that parasitize cutworm caterpillars, and one of them has larvae that parasitizes bugs, including squash bugs. So these are great pest control on farms, and the fact that the farm plantings attract them in large numbers works both to the flies' benefit and to the farm's benefit. Lepidoptera, the moths and butterflies, also did really well in our data. We detected almost a third of the butterfly species that are known from Martha's Vineyard, which is pretty amazing considering the small spatial area we were sampling and the limited habitat diversity in that area. Lepidoptera, butterflies, and a lot of the moths have, like this skipper in the bottom here, a really long, flexible proboscis that they can control like an elephant's trunk with a lot of dexterity. And that allows them to feed on all kinds of flowers, including things that other insects really are unable to feed on. For example, those baseball-sized, highly doubled zinnia flowers that are really popular as an ornamental. Almost nothing visited them except for butterflies, and you'd see butterflies stomping around on them with their proboscis going, sneaking down into the flower and finding nectar. Atalopetes huron is an interesting butterfly. It was the most commonly recorded butterfly in this project. And it's noteworthy that the first record in Massachusetts for this species came less than 30 years ago in Chomark, as it happens, in July 1997. It is a species that is expanding northward with climate change and has gone from being non-existent in Massachusetts to being really common in warmer areas of the state. So one thing this study did was help document changes that are taking place as a result of climate change. And we found a couple of pretty good butterflies in the sense of being rare or at least uncommon on Martha's Vineyard. A cloudless sulfur is one of them. It's a migratory species with a southern distribution that most years shows up here in small numbers as a migrant. Some years it's fairly common, but most years I see just one or two. And we did have one, I think that was at, I don't remember what it was, I think it was at Thimble Farm. And then we also had a juniper hairstreak, which is a very specialized resident butterfly. Its larvae feed exclusively on eastern red cedar. The adults, of course, will visit all kinds of things, but they rarely stray far from their larval host plant. So that was a good one to find. The distinction of butterflies and moths is really kind of an arbitrary one and doesn't really make much sense, but it seems to be common usage, so we observed it in this study. And we found a lot of diurnal moths, some of them like the Ailanthus webworm moth Ativa aurea in the middle there on the bottom, was very, very common late season moth, particularly on goldenrod. The Hawaiian beetworm moth over there on the left was a really good example of how our multiple ways of collecting data turned out to be beneficial. That moth was wildly common sometimes on farms. You would see hundreds of them, but they're very, very difficult to photograph. You flush them and they disappear. They perch on the underside of a leaf where you can't get the camera pointed at them. But we captured that, so we might get only one or two pictures in the course of a site visit. but our site visit narratives were able to capture the prodigious numbers of those that we saw. So that was how our different data sources worked together. And then Spraguea leo, I don't think it has a common name, is a certifiably rare moth on Martha's Vineyard and in Massachusetts. We are told that there is only one recent record from elsewhere in the state, so finding that was a useful contribution to moth studies on the vineyard that this project made. Onto the Hymenoptera, the wasps and bees. And this is a group that the wasps that are probably underappreciated. For some reason, people just don't pay much attention to them. Very few of them will actually sting you. Only a very small number of social wasp species are at all aggressive, and even those are only aggressive around the nest. So give wasps some love. They're actually ecologically very important and very interesting in their biology. We recorded a lot of individual wasps and a very high diversity of species from the 8 participating farms. A couple of the more common ones were the northern paper wasp, which is one of the social wasps, and the fourth-tooth mason wasp in the right there, which is a solitary wasp. And like most of the solitary wasps, individual females build their own nest, and they provision it with prey that they've stung and paralyzed, and then they seal it up, and the eggs that they've laid in those nests hatch and consume the prey items that the adult female has stocked the nest with. So wasps, particularly those solitary ones, are very, very important for population regulation in the natural world. And the solitary wasps, like Polistes, are predatory ones and scavengers. So they also serve important ecological functions on farms and indeed everywhere else. 2 other wasp families that are ecologically important and turned up in large numbers in this project were the spheccid wasps and the cribronid wasps. There really aren't common names that do justice to those families. But again, these are families, most of the members of which are predatory in terms of supporting their young, but vegetarian in terms of supporting themselves as adults. And again, goldenrod and spotted bee balm were magical plants for attracting these wasps. They're beneficial wasps. They serve important functions on farms and in the greater ecosystem. And these were 2 plants that this project demonstrated are very, very good for supporting those wasps. And then we found a couple of, I think, significant finds. For example, a Scalia bicincta that turned up at Morning Glory Mermaid Farm towards the end of our project. And again, note that it's on spotted bee balm. It's that magic plant for wasps. This is only the 2nd record in iNaturalist from the Cape and Islands region, and I have to plead guilty for accounting for the first one also over in Eastham on Cape Cod a few years back. But it's a very, very rare wasp. I don't know why, but it's a rare wasp on the coastal plain Cape and Islands. And the first vineyard observation came from this project, as I said. And then Cleonymus is another genus that is infrequently reported and actually extremely beautiful. This photograph doesn't quite do justice to the really metallic color that this genus has. But again, that was a pretty significant find that came out of this project. Onward to the bees, which are my real love. And I'm going to check the time here to see how doing okay. All right. And we'll start with the genus Bombus, the bumblebees. And they're represented by about 11 species, current and historical, on the vineyard, of which we found about 5 in the course of this project. They're social bees like the honeybees. They form colonies that a queen founds. And in general, they are generalists in terms of their flower visiting behavior, with both males and females visiting a lot of flowers, different kinds of flowers. And the most commonly recorded one was this one, the common eastern bumblebee, Bombus impatiens, which is a wildly common insect on Martha's Vineyard with a long season. It was strongly represented in this project. And we documented it visiting at least 46 flower species out of probably, I don't know, 80 or 90 flower species that we found things on. So it's a real workhorse pollinator. It probably does more pollination on farms than any other species. And just an important ecological force everywhere on the vineyard. And I should tip my hat to Caitlin Jones at Mermaid Farm, who found a couple of Bombus impatiens nests in decaying hay bales, which is a useful thing to know if a farmer is interested in supporting bumblebee nesting. Leave a couple of skanky hay bales out on the landscape, and you are likely to get bees nesting in them, bumblebees nesting in them. Interesting situation on the case of these two species. They don't really have good common names, and they're functionally indistinguishable in the field and from photographs. I can't even tell them apart from specimens. But so we lump them together as sort of a two species group. And interestingly, all of the, we recorded them at all 8 of the farms, I think, and that species group. And interesting, all of the iNaturalist observations of this species group from Martha's Vineyard come from farms. And I can say that in spite of having now about 2,400 bee observations in iNaturalist and a collection that I curate of 650 bee specimens and thousands of bees that I identified at least tentatively in the field, I've never found either vagans or Sandersoni anywhere other than on a farm. And I don't really know why this association is so clear. It's not an absolute association. That Goldstein's study that I mentioned did find Bombus vagans in non-farm habitats. But the data are really convincing that there is something specific about farms that these bees really like. And it may have to do with clover, which we found them on pretty regularly. And it may have to do with nesting sites. You know, maybe they like to nest in big compost piles, or maybe they like to nest in rotting hay bales. But for whatever reason, this project indicated that the farms on the vineyard are probably playing a very major role in supporting these two uncommon bee species on Martha's Vineyard. And Pepinapis, I tip of the hat to Molly Jacobson for this crippling photograph. Xenoglossa pruinosa, or the squash bee, was one of my favorite bees. That turned up on probably 6 or 7 of the farms, I think, invariably in association with large squash blossoms. And that's what these bees do. They live around squash. They collect nectar and pollen from squash blossoms. And they like the really big blossoms, you know, not cucumber flowers, but winter squash and pumpkin blossoms. So if you're growing pumpkins on a farm, keep doing it because these bees love it and you're helping them out. The genus Andrena is a very important genus, well represented on the vineyard with about 40 species. And we found almost half of those in the course of this project, which I thought was a pretty remarkable number and suggests that farms offer generally good habitat for this genus. The genus includes both generalists that will visit pretty much anything, and also a lot of specialists, in particular some that are specialists on late season composite flowers like goldenrods and asters. And I should say that this kind of specialization is common in bees. Probably 20 to 25% of bees could be considered specialists in terms of their foraging habitats. And usually it's the bee that is the adjuster, the adapting species here, that some species of plant is producing pollen. And over the course of many generations, certain bees develop adaptations that make that species particularly good at exploiting the pollen from those plants. So they may have specialized hairs for carrying it. The pollen, they may have, the larvae may have enzymatic adaptations for digesting the pollen. Anything can, a bunch of mechanisms. But with a lot of these specialists, you really won't see them on any other plant other than their favorites. And so if you don't have those plants, you don't have those bees. And that was one of the things that this project was really focused on exploring on farms. It is worth noting that some specialized bees that we expected to find did not turn up. Andrena bradleyi, which is an early season blueberry specialist, went unrecorded. I had really high hopes for a tremendous blueberry patch at North Tabor Farm and for some really wonderful high bush blueberries that grow along the road at Mermaid Farm. Those flowers supported a lot of things, but they didn't support Andrena bradleyi. So you win some and you lose some in the insect hunting game. Just a few of the examples of the Andrena species we found. Andrena hirticincta in the middle there is a goldenrod specialist, easily recognized by these bold hairbands. Andrena quitegi and Andrena tridens turn up mainly on woody plants, although quitegi obviously uses some herbaceous ones as well. Tridens turned up in large numbers on a crabapple tree. I mean, large numbers, I mean 40 or 50 at a time on a single crabapple tree at North Tabor Farm. Say a little bit about this tiny little bee, Andrena melanochroma. I found it on strawberry pretty regularly in the course of the project. And I did some research, and it turns out that people have studied the effects of native bees on pollinating commercial strawberries. And the result of this study that I reference here was that the native bee associations did much better at pollinating strawberries effectively than did native honeybees. And the mechanism for that was really kind of cool. You can see what a strawberry flower looks like, that picture at the bottom with the stamens sticking up above the rest of the flower. And it is an interesting fact that strawberries are able to both self-pollinate and cross-pollinate, but when they self-pollinate, the resulting fruits are usually smaller and not very well shaped. So they're less valuable from a commercial perspective. And when honeybees land on a strawberry blossom, they're big bees, they stomp around clumsily, and they knock a lot of pollen off, and it drops down, and those flowers self-pollinate. So they don't produce very good fruit. Whereas tiny little bees like Andrena melanochroma, the females are only about 7 millimeters long, and the males are smaller, get in under the stamens and pick up pollen on their bodies, but they don't really trigger as much self-pollination. They're much better at cross-pollinating strawberries. So strawberry crops that are pollinated by these native bees produce better fruit on a consistent basis than do strawberry patches that are pollinated by honeybees. And speaking about honeybees, we looked at the question of competition between honeybees and native species. There was a growing body of literature that demonstrates pretty convincingly that, at least under some circumstances, honeybees, when they're an introduced or non-native species, can compete pretty aggressively against native bees, and to the point of impacting native bee populations in a negative way. We witnessed a lot of examples where honeybees bumped native bees off of flowers, which doesn't seem like a big deal, but it mean that the native bee experiences a cost in terms of energy it needs to fly and foraging time. It loses a little bit of time. So if they're getting displaced from flowers over and over and over again by honeybees, that could potentially add up to something that would impact those bees' ability to survive or to reproduce successfully. So while we think honeybees are wonderful and amazing animals in a lot of ways, We don't really like them from an ecological perspective because they're non-native and potentially destructive competitors in the broader ecosystem. But our focus was on feasible improvements that farms could make, and some farms, and we can't complain about this, choose to grow honeybees, host honeybees, either for pollination services or for honey as a crop. And there is nothing wrong with doing that. So we looked for ways that we could offer that would reduce any potential impacts. And the thing that we really came up with was by simply making sure that you have a lot of varied floral resources at all times during the season. Because what happens then is what we call resource partitioning, where different kinds of bees will go to different kinds of flowers. Really good example here in May at Thimble Farm, where a mixed species cover crop was being used by 2 bee species. The honeybees were all on crimson clover, and the Bombus griseacalis were all on vetch. We watched for about 20 minutes, and we didn't see any interspecific interactions at all. They simply divided up, they visited their favorite flowers, and went happily about their business. Another example, late in the season in Thimble Farm, where we found a whole bunch of honeybees, hundreds of them, feeding on a row of kale that had bolted and gone to flower. And they were evidently taking nectar from the kale blossoms. And we found nothing except 1 carpenter bee and a whole ton of honeybees on that kale. Meanwhile, at the project plot, we found some Lasioglossum, a native bee genus, and a native Bombus impatiens, but no honeybees. So again, the bees had kind of divided the landscape up and were visiting resources that they particularly liked and staying out of each other's way. So we think that if farms, or for that matter, individual people who choose to host honeybees, can keep enough variety and enough quantity of resources available through the full season, we think it's pretty unlikely that there will be significant negative impacts on native bee species. So that was the best we could do on the basis of our the analysis that this project supported. We did find a couple of significant bee records, first county records from this project. Andrina bisalicis on the left and Megachile inimica on the right, taken off of Black-Eyed Susan at Thimble Farm. First county records are unusual. I mean, we get 1 or 2 a year, but there have been 204 bee species now documented on the Vineyard. So it isn't every day that you find a new species. And again, this is an indication that farms are good enough habitat for bees so that even pretty rare things turn up there. And that was good news that came out of this project. So did it work in terms of enhancing pollinator resources on farms? Well, yeah, it did. There were some qualifications to that. And I talked earlier about the importance of landscape context, and I want to take a little bit of a deeper dive here. Solitary bees only disperse 100 meters, 200 meters on average from their nest sites in order to collect pollen. So if you don't have a population of a particular bee within that distance of the pollinator patch that you've created, you're unlikely to get that bee. It will turn up only very, very rarely or not at all over time. Simply because you're out of the range, the bees are not going to find it because they're not traveling far enough to discover the resource that you've created for them. So in a situation like the project plot at the Farm Institute, we've had plenty of goldenrod. It did very well in the project plot, and a lot of generalist pollinators visited it, but we didn't find any specialized goldenrod specialist bees there. And that probably was because there simply weren't any close enough by to discover that resource. There is goldenrod down by South Beach, and you find a lot of goldenrod specialists down there. but that's too far away. They just don't range far enough to find the goldenrod that we put in at the Farm Institute. And if that's the situation, then you're simply not going to do much good by putting in goldenrod. You're not going to support those specialist bees with that choice. So you maybe want to look at other things, putting flowers that support other taxa, or thinking about providing nesting habitat for bees or something like that. Or maybe you want to think about a project that creates a lot of goldenrod patches around the entire landscape at a much greater scale to sort of connect things better for those specialists. But you need to find a different strategy if you haven't got populations of those bees nearby. In some other situations, there may be so much of a resource that adding a few flowers doesn't really make much difference. And I would point to Mermaid Farm as an example of that. Mermaid Farm is just loaded with goldenrod, and the small amount of goldenrod, the 2 5-by-5 foot 50 square feet of goldenrod that we put in was really just a drop in the bucket. We did find some specialized bees on our plants, but we found a lot more on the goldenrod that was pre-existing, simply because there was so much of it there. So again, in that case, there's probably no point. I mean, you're not doing any harm by putting in goldenrod. But if your goal is to support rare bees, you're not really doing much good by adding coals to Newcastle here by putting in additional goldenrod. But in between these two extremes, there is this sweet spot where there is enough pre-existing resource in the general area so that you have populations of some of these specialized bees that are kind of nearby. They may not be visiting the farm because there's no goldenrod there, but they live close enough so that if you put in goldenrod, the bees will discover it, and then maybe they'll nest nearby, and you're going to end up adding additional habitat and additional nesting area and different resources for those bees. And that's really what we were trying to accomplish with our project plots. And we didn't really anticipate how important the landscape context was going to be. But that was one of the take-homes from this project, was that for farms that are in this sweet spot, you really can make a difference in terms of supporting uncommon and specialized bees by putting in even pretty small appropriate plantings. And you can make more of a difference by putting in bigger appropriate plantings. Here's an example, it's just a specific example of that. Andrena nubecula, beautiful little bee, sometimes called the cloudy-winged minor bee because of these pigmented wingtips that it's got. It is a resolute goldenrod specialist. I don't think I've ever seen it on anything else. We didn't find a lot on in the course of the project. It's not a terribly common bee on the vineyard. We only found 7 individuals on 5 visits across 3 different farms. And in one of those cases, it did turn up on goldenrod outside our project plot. But in the case of the other 2 farms, where all of the other instances of Andrena nubecula occurred, they were on farms that didn't have any pre-existing goldenrod, but we were able to locate goldenrod within a fairly short distance. So there was pre-existing goldenrod around the farm, and there were pre-existing populations of Andrena nubecula using that goldenrod. And when we added goldenrod to the plantings on the farms, Andrena nubecula discovered that resource and exploited it. And that's exactly what we were trying to achieve. And this is not a lot of individuals, but to me, it's a pretty compelling signature of our project plots doing what we had hoped they would do. Just to look at some of those late season composite flower specialists, 2 of them, Andrena asteris and Andrena hirticincta, were very, very common. They turned up at most of the farms. Andrena asteris, most of those records came from a plant called Symphyiotrichum pilosum, and most of that was at Thimble Farm, where that was a pretty common plant in the area of our project plot. And then Andrena hirticincta is a very common goldenrod specialist on the vineyard. So we weren't surprised to find that at lots of places and using lots of different resources. But look at the pattern with all of these other less common specialists. And for the most part, they turn up in the project plots and not in the general area around the farm. So again, the project plots were an added resource that these bees were finding and using. And they weren't using pre-existing resources on these farms. So it's again, a small number of species. Statistically, I wouldn't really want to insist on this too much. If we did a statistical analysis, it may not rise to the level of what biologists call statistical significance. But looking at it practically as a naturalist, I have no doubt at all that our project plots made a difference in this general idea of putting in specialized the resources for specialized pollinators, if it's done in an appropriate landscape context, can make a difference, and even a significant difference in some cases for these rare and specialized bee species. So in that sense, the project was really a success. It was also a success just in terms of producing a really huge data set and producing a really good survey of the diversity of insects that were using flowers on farms, a good diversity of both taxonomic and functional groups, with particularly the orders Lepidoptera, Diptera, and Hymenoptera. That's moths and butterflies, flies, and bees, wasps, and ants, and sawflies. All exhibiting really high diversity. Our project plantings met their goal, at least in some cases. All the effects were sometimes subtle and were generally conditioned, both in terms of the number and the species composition, by the nature and the extent of pre-existing resources around the farms and on the overall landscape contexts. So that was an important message that this project came up with. And finally, again, goldenrod with a side dish of native asters. We didn't experiment with woody plants because we didn't want to put basically a permanent thing in on these project plots, and also because woody species take a long time to mature. A lot of the other pollen specialist bees on the vineyard use woody plants like blueberry and lyonia and things like that. So we didn't deal with those in the course of this project, but we did deal with these late season composite specialists. Goldenrod attracted them reliably, asters attracted them reliably, and they also attracted a lot of other kinds of more generalist pollinators. So the more goldenrod and the more native asters that you put in any context on the vineyard, I can't imagine a scenario in which you're doing harm by doing that. And I think in any circumstance, you're benefiting at least the generalists, and in many cases, also the more specialists. So I'll say it one more time, plant goldenrod, it's wonderful stuff. We hope to encourage both farms and indeed other people on the vineyard to adopt some of the recommendations and the strategies that this project experimented with, based on the demonstrated importance of late season composite species. We would love to try to find a way to develop a good, robust local propagation source for that. If any farm wanted to take on like the commercial scale production of 6 or 8 goldenrod species, I bet you I could find some grant money to get you started. So think about it. It'd be really beneficial. And we hope to encourage natural neighbors, participants, and other BiodiversityWorks program to implement some of the strategies and to plant some of the plants that we found were successful in the course of this project. Thanks to a lot of people who helped develop the project, help run it, help identify bees, and especially to the 8 farms. It was really an honor and a privilege to work with these farms. Wonderful cooperation, wonderful places where, as I said, I really enjoyed poking around. They're doing great work. They're both providing sustainable food for the Martha's Vineyard human community, and they're providing wonderful resources for the broader ecological system of Martha's Vineyard. And really, really grateful for their participation. And of course, as I always do in these presentations, I would like to thank the insects for existing and for being extremely cool. And that's what I've got for you today. If there are questions that I could answer, you're welcome to unmute yourself and fire away. And if I don't know the answer, I'll make something convincing up. How's that? Anybody?
Matt, is there anything you would add to the studies if you did these again, or things you would do differently?
Yeah, I would work with somebody who really knows about data management to improve our data collection and data management methods. The data that we have in those site visit narratives is enormous, and it's really good data, but it's really, really hard to get at. It's like if you're looking for something in particular date, a particular farm, you can go there and you can find what we saw. But if you want to say, well, how many observations did we have, or how many individual Bombus impatiens did we find? You would have to go through 8 years, three years of 8 text files, searching for Bombus impatiens and adding up all of those numbers manually in order to get that count. And so we were really, really limited in some kinds of analysis that we could do. The data are all there, but I wish we could have found a way to make that, to get that same information collected, but in a way that made it easier to massage. That's one of the main things. I also wish we could have experimented with some woody species like Amelanchier or blueberries, vaccinium. We've had good results elsewhere. Nancy, I see your smiling face there. Do you have a question? No, you're just smiling. Okay. Anybody else? Then I think we're done. I will let you be on your way to your dinner and your other evening activities. Again, thank you so much for participating with us this evening. I really appreciate your taking the time and showing the interest to show up. I hope this was edifying. I hope you will plant some goldenrod, and I hope you enjoy the rest of the season and find some interesting insects. So thank you very much.
Okay. Thank you, Matt. Loved it.
Yeah. Thanks, Matt.
Thanks, Matt.